Biophysics applies the theories and methods of physics to understanding how biological systems work. Biophysics is essential in looking at how the molecules of life are made, how cells move, and how the complex systems in our bodies function. Understanding how all life works requires experts from a wide variety of fields, and it takes a vibrant interdisciplinary community to make that happen. Now, in San Diego, California, scientists from all over the world are coming together to share, learn, and discuss the very latest in biophysics. This is the 2020 Biophysical Society Annual Meeting, and we're Biophysical Society TV. It is day 2 of the 2020 Biophysical Society Annual Meeting, and as symposia are gathered and lectures take the stand, we're here to make sure you know what's not to miss this year and to get an insight into some of the most incredible research happening in biophysics today. Well, if you've not already, join us here at the Convention Center or at your hotel room at the Hilton or Omni, or online on the meeting website or the app. Don't miss what we've got in store.
It turns out if you really want to understand what's happening in the body or what goes wrong in the body, you need to understand the rates or what we call kinetic stability, how fast or how slow processes take place.
What we're really lacking is a mechanistic understanding of how the machines that support the replication life cycle, how they function. And that's where biophysics really comes into play.
You can imagine that a cell might be stretched, or there's physical inputs coming from the matrix that they're attached to, or there might be shear sensing or flow. So what we're interested in is, can we apply these different mechanical inputs and see whether different mechanically activated channels respond to one over the other?
What makes this such an exciting place to be?
Because there are so many good scientists and then they are talking about their new results. Nothing could be more exciting than that.
Now, the work done in biophysics can have a major impact on people's lives all over the world, and this couldn't be more apparent than in research being undertaken to understand the mechanisms behind the Ebola virus. And to better understand this, I'm joined by this year's Future of Biophysics lecturer, Professor James Monroe. Thank you so much for joining us.
My pleasure.
First, I'd like to know, how well understood is the Ebola virus today?
So what we have is a very general understanding of the virus. We understand the major landmarks that the virus encounters when replicating. We know what the virus looks like. We know the components of the virus. What we're really lacking is a mechanistic understanding of how the machines that support the replication life cycle, how they function. And that's where biophysics really comes into play.
What do we know about how the virus gets into human cells, and why do we need to know more?
So we know that it enters by way of fusing the viral membrane with a cell membrane, and this process is promoted by a protein that's found on the surface of the virus engaging a protein on the surface of the cell, and this leads to a cascade of events that fuses the virus with the cell, and that by doing so it empties the contents of the virus into the cell and initiates replication. Why we need to know more is because this is a historically underutilized drug target for all viruses. And it makes a good intuitive sense that you would stop infection at the point at which it begins.
And that's incredible to hear. What's single molecule threat then, and how are you using this in investigating how Ebola does this? And secondly, what does your experimental process look like?
So single molecule FRET is a method by which we get dynamic insights into how the molecular machines that support virus replication work. How do they change their structures as they go through this process? What are the binding events that occur between the virus and the host cell? And we get all of this at a single molecule resolution. And that is exactly what it sounds like. We can see these events occurring one molecule at a time. So in a sense, we end up with a trajectory of how a given protein goes from how it starts on the virus outside the cell into what it looks like after it's entered the cell.
And as far as your experimental process?
So this is a fluorescence-based approach. So what that means is that we need to attach a fluorescent dye molecule In fact, two fluorescent dye molecules to our protein of interest. Okay, in this case, it is the envelope glycoprotein from the Ebola virus. And then it needs to attach to sites on this protein that are going to be informative, that are going to inform on the dynamic events that we're interested in. So that's the major bottleneck, is figuring out where we're going to attach the fluorophores. Once we've crossed that hurdle, it's a matter of purifying the virus, labeling it with these fluorescent molecules, and then immobilizing the virus on a surface that we can image with a microscope. And everything downstream of that is data analysis.
Is it dangerous?
Is it dangerous? It's no more dangerous than any other study of proteins or nucleic acids in biology. We're able to form particles that only have the components that we want to study, and we leave out everything else. So it can be handled in a normal laboratory environment.
What exactly can you learn from this research, and how can this research lead to breakthroughs in creating vaccines in Ebola and other viruses?
So what we get from these experiments are an accounting of all the different structures, or all the different shapes that this molecule can adopt. And we can see then how the distribution of different structures changes after the virus has interacted with the cell. So the question then becomes, can we then use what we've learned to engineer this protein to look more like the structures that the antibodies recognize? Because if we can do that, then we can form a better immunogen that goes into a vaccine and can more efficiently elicit antibodies that will be protective.
Incredible work happening at the University of Massachusetts with Professor James Monroe. Thank you so much for joining us. Appreciate it. Well, I can't wait to see where Dr. Monroe's research goes next. But now back to the meeting co-chairs who tell us again what's not to miss at the meeting. Let's take a look at Monday's program.
So the symposia on Monday include two that I'd like to feature here. One is Biophysical Underpinnings of the Origin of Life. Many people are familiar with the RNA world hypothesis, but in this symposium we're going further, talking about how, for example, nucleic acids might have started, but then a transition to a more protein-centric world.
For Monday, I'm really excited about the sensational membrane proteins, and these are membrane proteins that help us feel mechanosensation when we're pinched or poked, membrane proteins that help us taste sour, and membrane proteins that help us be able to smell.
The other session I'd like to highlight is the kinetic stability session, where we're looking at non-equilibrium thermodynamic systems and how kinetic stability, for example, might enable a protein that isn't perhaps particularly stable to persist for over the lifetime of a cell.
One of the more exciting symposia is the Futures of Biophysics Society. And these are 4 young investigators nominated by the society at large, doing outstanding work. They're the up-and-coming risers of our field, and it should be, I think, a really exciting session that both Patricia and I will be co-chairing.
Yes.
So I definitely recommend that everyone go see the Future of Biophysics.
Now to get a preview of one of this week's carefully crafted symposia, I'm joined by Chair Professor Jonathan King to learn more about the themes behind it. Thank you so much for joining us, Professor King. First, tell us about kinetic stability and what's the overall focus of the symposium?
Now we're at a point in biophysics where we're beginning to bring the basic biochemistry into the question of human disease, human pathology, what's actually going on in the body. And inside the body, proteins are in a very different environment. You know, if you think about a muscle, those myosin or tropomyosin or troponin molecules, they're not in solution. They're in some very dynamic interacting state, interacting with small molecules, big molecules. And it turns out if you really want to understand what's happening in the body or what goes wrong in the body, you need to understand the rates or what we call kinetic stability, how fast or how slow processes take place. And that requires a different measurement than just the equilibrium.
What exactly will be covered in the session?
We'll be covering the range of proteins that show this big difference between thermodynamic and kinetic stability. The molecular determines what are the interactions, amino acid interactions inside proteins that give it, or features give it this kinetic stability. Its relationship to this very important class of diseases, amyloid diseases, and also that leads to the notion, well, maybe we can prevent the accumulation of some of these most destructive forms of the protein, the amyloid fibrils, by, if you understand the pathway by which they form, you might be able to intervene with small molecules.
Which is always the most important,
Yes.
right? Being
Yes.
able to intervene.
Yes.
Can you show us on the screen, please, with some of your own work, how you're looking at the cataract?
Well, here's a diagram of the eye, and in the center there is the lens, which has to be transparent so that light passes through. Lens is a very distinct tissue. It's packed full of a distinct class of proteins called the crystallins. This is true in all lenses of all vertebrates, marine mammals, etc. And I'm going to talk about the gamma crystallins, which are Greek proteins. And for every person out there who's looking at this slide, you're seeing it through a population of crystalline molecules that was synthesized when you were in your mother's womb. So these proteins have to remain folded and stable all your life. And if they don't, if they slightly unfold and those things stick together to form a very stable aggregated state, then you get cataracts. We've now been able to identify, well, exactly what are the forms of damage that lead to this state, at least for damage due to ultraviolet light, which is one of the major etiological causes of cataract in humans and in animals.
How do you think your work is going to help people ultimately?
If you have millions of people getting cataract surgery, the complications are in hundreds of thousands. Many people are not able to get cataract surgery. And so we feel that if you really understand this pathway, as we're coming to do, you ought to be able to interfere with it with small molecules, which could be applied as eye drops, and ought to be a very effective way. The first such small molecules are coming into clinical trials. So we hope for humans in the years to come, it'll be possible to slow down or prevent cataracts in older people.
Well, so much to look forward to for so many of us who suffer from this. We don't even know it, but we do. Thank you so much for joining us, Professor. Really appreciate it. Now across the Atlantic to Saarland University. That's where researchers are combining theoretical and experimental modeling to look at the energy dissipating dynamics in cells and molecules.
20 years ago, we started biophysics at Saarland University, and meanwhile, biophysics became a focus area of the physics department, but also of the whole university. And that's why we decided in 2016 to found the Center for Biophysics. The Center for Biophysics consists of people from material science, life science, the university hospital, and this kind of interdisciplinary approach is also characteristic for our research projects. And we also want to train our students in this interdisciplinary way of thinking, and that's why we implemented a bachelor and master program in biophysics.
The Collaborative Research Center CRC 1027 is the nucleus of the Center of Biophysics, established in 2013 as an interdisciplinary research initiative between biophysics, biology, biomaterials, and medicine. Our scientific aim is to reveal and understand physical principles governing the spatio-temporal organization of living active matters into molecules, cells, and tissues. An important purpose of a collaborative research center is structural development of the home university, which is in our case the establishment of a new building for the biophysics, which will be finished in 2024. Another very important aspect of a collaborative research center is the support of early career researchers and equal opportunity in research.
We are interested in biofilm formation. To study bacterial adhesion, we use modern surface science techniques such as photoelectron spectroscopy, ellipsometry, scanning and transmission electron microscopy, or atomic force microscopy. We attach one living bacterial cell to a tiny cantilever and press it onto a surface and measure the force that is needed to detach the cell. The proteins that are part of the bacterial cell surface mediate adhesion. Type and density of these proteins determine the adhesive strength of bacteria. We even produce our own samples of test teeth. Our colleagues from the dental clinic test our artificial teeth and compare the results with real teeth, as well as the impact of plant extracts or appetites.
I'm in the Center of Biological Physics and I'm interested in cooperative effects, and what we're studying are DNA interactions, symmetry breaking in the embryo, the incorporation of non-canonical amino acids during transcription translation. And I think as physicists and at this interface in the center, we have sort of all colleagues which understand and can help in what we're doing, technically speaking, but also in terms of ideas. And especially with the new building that we're going to get, I think we can make a very strong team and sort of attack these kinds of subtle problems, which on the long run will be extremely fundamental and important.
We are here at a collaborative research hub between Saarland University and the Leibniz Institute for New Materials in order to tackle biophysical questions. We're specifically interested in the cytoskeleton. For example, we look at the actin cortex, trying to understand how the mesh size, which we look at using electron microscopy, alters cellular mechanics. Another example is where we look at how vimentin influences actin polymerization in stress fibers. And here we really have the good possibility to use interdisciplinarity, where we can go really from the far end of biophysical simulations to really look at single molecules up to having real cells from real patients coming from the clinics. And this is an opportunity we are really happy to use. We are also happy that we are able to take these tools which we use to really tackle our scientific questions and bring them directly to the student, so that they can have a very early hands-on experience and try these tools.
During my bachelor thesis, I was already working in a research group here, and I feel that especially, or even for bachelor students, you can really already see how the research is conducted here. I think it's really exciting to use very expensive and very new equipment, even for a bachelor student. I have now experience with different types of microscopes, and you are able to look into very different research fields.
Within the Center of Biophysics, we study how cells sense and control the properties of membranes and how abnormal properties of membranes lead to complex metabolic and neurological diseases. We are a group of computational biophysicists, and our goal is to combine experimental data with computer simulations. And we do this to help experimentalists to learn as much as possible from the data, and on the other hand, we aim to make our simulations as realistic as possible. The Center of Biophysics provides a fruitful environment for us because it integrates a range of methods, from very theoretical methods over computer simulations to experimental model systems up to studies on the whole cell. And it's this integration of methods that helps us to tackle modern challenges in biophysics.
We are in the Center for Integrative Physiology and Molecular Medicine. Our main question is, how can the immune killer cells kill the tumor cells efficiently? And the second is, how is this killing efficiency regulated by external factors? So to address those questions, we need not only the expertise from the immunology, but also it requires the expertise from many different fields like the theoretical physics and also bioinformatics and material science. This interactive and interdisciplinary milieu is actually not easy to find elsewhere, to have everything packed under one roof. So that makes the Center for Biophysics very unique for us.
We are interested in biofilm formation. To study bacterial adhesion, we use modern surface science techniques such as photoelectron spectroscopy or atomic force microscopy.
Our center research focuses on the macromolecules and machines of life. This goes from nanoscale assemblies all the way to cells and how they organize to form colonies and differentiating tissue.
For us, we welcome new users
And that way we bring all the community users together and we together discuss about their research leads.
As scientists, we have to make knowledge available to the broader scientific community. This is really at the core of what university research is supposed to do.
Now to talk about sensation membrane proteins. They play a key role in how our cells sense their surroundings. But how does this process work? To walk us through this, I'm joined by Dr. Kate Poole. You're the symposium speaker here. Thank you so much for joining us. The first question is broad. What are sensation membrane proteins and where are they found? Also, why are they so important?
So the sensation membrane proteins that I'm interested in are ion channels that are actually activated by mechanical inputs. And they're found in a surprisingly diverse range of cells and tissues. So the ones that are easy to imagine are those that sit in your somatosensory system and help mediate our sense of touch and pain. But they're also found in other mechanosensitive cells and tissues, such as the chondrocytes, which are the cells in your cartilage that need to sense and respond to changing mechanical loads, but also cells like melanoma cells that can actually sense changes in their physical surroundings as they break away from tumors and migrate through the body.
That's incredible. How does mechanosensing vary in cell types, and can you give us an example?
So this is one of the things that we're actually really focusing on. The question of how can cells actually sort out all of these different mechanical inputs. So you can imagine that a cell might be stretched, or there's physical inputs coming from the matrix that they're attached to, or there might be shear sensing or flow. So what we're interested in is, can we apply these different mechanical inputs and see whether different mechanically activated channels respond to one over the other?
What molecules specifically are you looking at and how are you doing this?
So we do, of course, focus on piezo-1, which is sort of the best characterized mammalian mechanosensitive ion channel, but we've been comparing that to other ion channels such as TRPV4, a new one that we think we've discovered, alkan-1, so we can't for sure say that this is a mechanically activated ion channel yet, but we've got pretty good evidence, and then also TREC1. And so what we actually do is we use two techniques that have been well established for a long time in the field to apply mechanical inputs. We can either indent the cell with a large glass probe, or we can stretch the membrane using our electrophysiology pipette. But what we wanted to do was actually add another technique where we could directly look at what was going on at that interface between cells and their surroundings. So we grow our cells on an elastomeric pillar array, and we can actually move individual elements underneath the cell. And so we've been asking, all of these different molecules, do they respond to all of these mechanical inputs, or can they actually distinguish between them? So when we look at piezo-1, we see that it actually responds to all the three types of stimuli. In contrast, TRPV4 is completely agnostic to what goes on when you indent the cell or stretch the membrane. It only senses mechanical inputs when it impinges the cell from this elastomeric substrate. With TREC, we see the opposite, so it doesn't actually respond to these substrate deflections. And with alkan, we get yet another profile where what it doesn't sense is membrane stretch. And we think that this is telling us that perhaps cells themselves have a tunable sense of touch.
Wow. What does it all mean?
Well, I think one of the things that it means is that this mechanosensation is complex. And it also suggests that in different cells and tissues, if you have more than one of these channels, it might be allowing those cells to actually integrate some of these different mechanical signals.
Lastly, when's the session and why should people go?
So the session is at 10:45 tomorrow morning, that's Monday morning. And people should come along if they want to learn more about these kind of channels. And there's other talks on piezo-1 as well, and thinking about smell and taste.
Incredible. Thank you so much, Dr. Poole. I appreciate you. Thank you. Well, now back to our favorite part of the show. Let's hear from you. You've traveled across the country or the world to be with us right here in San Diego. So what makes this meeting so special? Why come to the Biophysical Society Annual Meeting?
So the Biophysical Society meeting is actually this big meeting in the field. So it's a chance to meet people from all over the world that does biophysics, to see where they're from, what they're doing, and where they're going. I think we have like 8,000 people this year. That might be the biggest conference that we have in biophysics.
There's a lot of good science going on. If you compare to a lot of other conferences, BPS really does have quality talks, quality sessions, and it just makes it an ideal place to be if you want to start new experiments or think of new experiments and collaborations.
There's so much going on here that you can basically walk from room to room, see what's listed outside, and find some interesting experiment that you never even thought existed previously.
I'm local from San Diego. I'm a second-year PhD student from Scripps Research, and I want to learn more advanced biophysical technique, including structural biology and a bunch of other spectroscopy methods.
I mean, this is a meeting, with a lot of really good, excellent biophysicists. And we also work on ion channel, and that's a big topic in biophysics. So, of course, we have to be here to see our friends and then to look what happened, in the field in the last year.
What makes this such an exciting place to be?
Because there are so many good scientists, and then they are talking about their new results. Nothing could be more exciting than that.
Just the interdisciplinary nature of it all. So you have mathematicians, you have physicists, you have biologists, cell biologists, all coming together. And I like that mix of people. You get to see people that you normally don't see.
Having all of that together in one place is really powerful for kind of deciding what we should do next, and then also for networking and establishing friendships and professional contacts that help us move on to the next stages of our careers.
Yeah, this is my first time at BPS, meeting people whose names I've read on a bunch of papers during prelims and grad school. It's really cool to actually meet them and actually hear them speak about their research.
Fellow first-timer, thank you so much. As we saw there, this meeting is a great place to be for everything biophysics related, of course. Well, that's all from us today, but the meeting is a long way from over, and we have one more information-packed show in store for you tomorrow. We talk about coupling molecular activation with Dorit Hanane and take a look at the symposium on exocytosis and autophagy. Keep watching this show any way you can. We'll see you tomorrow.